Hi, everyone. Happy Friday. I'm Tanya Stevenson, the CEO of the Myotonic Dystrophy Foundation, and I am delighted to welcome you to our November Meet the Drug Developer session. It's my great pleasure to introduce and host Lupin Neurosciences today. Following a short intro by MDF, Lupin's going to present their progress of the development of a therapeutic for myotonic dystrophy. We will moderate questions after their presentation, so please send in your questions in real time while the presenters are speaking. About MDF, we envision a world with treatments and a cure for myotonic dystrophy. Our mission is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. MDF provides resources and support for families and healthcare providers. Toolkits and publications, including handbooks and clinical care recommendations, are available on our website. Our support programs, with nearly 30 support groups and over 40 support group facilitators, we welcome people from across the globe and have support groups in 3 different languages. Most are held in a virtual setting, so you can participate from the comfort of your own home. All of our events and activities can be found on the calendar of events on our website. And our digital academy is our library of past presentations and videos, including past conference sessions. They are all hosted on our website under the Digital Academy and available to everyone. Today's session will also be posted to our Digital Academy next week, and we will definitely send out an announcement to everyone about that. We hope you have all saved the date already in your calendars for the 2025 MDF conference in Indianapolis, Indiana. This is going to be May 2nd through 4th, but please note that the professional track is actually going to begin on May 1st with a Pharma Day with Urodyma and a cardiac endpoints workshop. Registration is now open for community and professionals, and that's myotonic.org/conference. We welcome you to watch our past Meet the DM drug developer sessions for details on the progress of other drug development efforts. It is on our Digital Academy, and you can find it directly on our website at myotonic.org/meet-dm-drug-developers. For MDF drug discovery efforts in general, and especially those entering clinical testing phases, are a culmination of our mission and help us hold on to the hope that change is near. Looking at our drug development pipeline, as you see here, helps, I think, to illuminate the tremendous progress that has been made in the last couple of years, as we now have dozens of companies focused on myotonic dystrophy, with 9 in the clinical testing phase. So clinical trial activities fall into 4 broad areas and follow the central dogma of biology. From left to right, you can see we have DNA harboring the gene in DM1 with the expanded repeat. In a process called transcription, the RNA is made, and RNAs are ultimately used in a process called translation to make function proteins. In myotonic dystrophy, the RNA harbors the expanded repeat and forms highly organized structures that ultimately function as a trap for the important regulatory proteins. So we often refer to those traps as hairpin loops. These regulatory proteins are not available. When these regulatory proteins are not available in our bodies, the cells and tissues and organs can no longer function properly, as we see in myotonic dystrophy. Drug discovery targets these 4 broad areas. So one is efforts focusing on the repeat in the DNA using inhibition of transcription or gene editing approaches. In 2, they aim to inactivate that toxic repeat. In 3, we try to compensate for those trapped proteins. And 4, we try to correct the downstream cellular defects that are caused. So today, Lupin, who we will hear from shortly, is focused on correcting that downstream cellular defect. And without further ado, it is my great pleasure to introduce the speakers from Lupin today. Klaus Jepsen brings over 30 years of leadership experience in the pharmaceutical industry, specializing in commercial strategy, portfolio management, and product launches across Lupin, Asia, and the United States. As president of global specialty at Lupin, he is focused on advancing the company's mission to provide access to innovative medicines for patients and communities worldwide. Before joining Lupin, Klaus led global product strategy for rare diseases at Takeda Pharmaceuticals, where he championed initiatives to deliver groundbreaking treatments for underserved medical conditions. Earlier in his career, he held various leadership roles at Ellipta in the US and managing Advair and Seretide in global markets. Also with us today is Mariska van Aswegen. She holds an honors degree in pharmacology, as well as a master's degree in business administration, specializing in executive management. She has more than 25 years of experience in the pharmaceutical industry in various roles, through which she has built extensive experience in developing strategies and resources to support disease management beyond access to pharmaceutical treatment. Over the last 7 years, she led the team to launch Lupin's first orphan drug in Europe, with a primary focus on understanding patient needs to support the development and implementation of research and development, and launch programs to improve myotonia management in people living with myotonic disorders. Also with us today is Dr. Alla Zozulia Weidenfeller. She has a PhD in cellular biology and has been trained in neuroimmunology in the US and in Germany. She currently holds a role of senior director and medical lead on the Lupin clinical program. With around 14 years of pharma experience in drug development, including target discovery, new drug launches in various therapeutical areas, as well as medical affairs, Alla has gained her experience in various scientific roles at EMD Serono, Alexion, and Novartis. Having spent many years herself in the lab before she has published over 30 peer-reviewed scientific papers and reviewed articles and book chapters and gained a strong record of expertise in pharmaceutical industry across all stages of drug development. Just a reminder that many of you have submitted questions when you registered. Thank you so much. Those questions were given to the presenters in advance, and they have done their best to integrate those answers into their presentation today. However, if you still have questions, you are very welcome to submit them live. Just click the Q&A button in your Zoom window here to open it. Type your response to the question in the text field. And you can also click the send anonymously box if you do not want your name to be attached to your question in the Q&A. But go ahead and type in your questions as we're speaking and click send, and we will get to those questions at the end of the presentation. Okay, it is my great pleasure to introduce the Lupin team. Welcome.
Thank you very much, and thank you for the very nice invitation and the introduction, Tanya. We very much appreciate that, and we are very happy to be here. We obviously are passionate about this area like you are, and therefore we hope we will spend an hour here in each other's company that will be really productive. I'll just very quickly introduce you a little bit to what drives us at Lupin. And if we go to the next slide, you have a little quote here from our founder. 50, 60 years ago, the company was founded. And it just occurred to me that your vision sounds a little bit like the vision that was laid out here by Dr. Gupta back in the time about a brighter tomorrow, a better future. And what we're really trying to do is we're trying to work hard on problems that are hard to solve, turn normal people into patients for at least a part of their life, and doing what we can to help ease the burden of being a patient. Ideally, we all want a cure. That's often a very tall order, but sometimes less can do it as well, and make progress on that agenda. If we go to the next slide. If we talk a little bit about who Lupin is, it started in India and in tuberculosis, which was a big burden of disease back 50 years ago. And it has evolved over the years, and today it's a global company. We supply a lot of generic medicine in the US and around the world, but we're also moving into more specialized areas, and what we're talking about today is certainly one of those areas. So we invest more and more in R&D as the years goes by, and we actually have a strong commitment to become a key player in rare neuromuscular diseases, and that's what we're going to talk about today. As you can see here, we have a number of other areas, as a lot of companies do have, but they're probably a little bit less relevant for today's conversation. And with that, I will pass it on for the next slide to my colleague, Mariska. Thank you very much.
Thank you, Klaus. The next slide is just to talk a little bit about the drug development process. And when we talk about drug development process, I'm sure you visualize the blue line that is represented here on the slide, which is a conventional process that takes on average about 15 years to bring an approved medicine with the data to support the use in a specific indication to the patient community. And talk about medicines, I think when we mention medicines, we refer to not just the chemical compound, the chemical entity. That's only the start of this whole process, but a medicine or a chemical entity becomes a medicine when you have generated the data to support the safe and effective use in that specific indication. When we think about drug repurposing, which is a different process that maybe starts a little bit earlier or later on the process, where you have already some data and some information that characterize the chemical entity at the start of this process, where you may have some data and some information of the safety of this chemical entity. And there may already be some existing signals of the efficacy of the drug in a specific indication. And that's where you may save some time in bringing this product forward into the process of generating the data that would support the safe and effective use of that product into a specific indication. The benefits of drug repurposing, therefore, is that you can follow this novel approach to bring a product to meeting unmet patient needs faster. And in some instances, it may reduce the time to market and to prescription of such a product once it is approved for use by the regulatory authorities by a significant amount of time. This is the process that Lupin is following in our drug development process that we are undertaking, that we will be sharing a little bit more with you later in the presentation. And if you go to the next slide, please. We also thought of sharing a little bit what our interests are in researching myotonia. This direction for Lupin and this research interest for Lupin is anchored in Mexiletine's approval for the treatment of myotonia symptoms in Europe in the patient group, the non-dystrophic myotonia patient groups. So we have an interest in generating evidence for myotonia management also in the myotonic dystrophy patient population. And our interest is also in understanding the patient needs better by engaging with the patient community, gathering insights from patients so that we can understand really what the challenges are for patients, for the myotonic dystrophy patient population. We have been gathering these insights in these engagements with the community and specifically in several patient surveys that Ala will be sharing the outcomes of some of the data from the INVA survey in the rest of the presentation. And then also we work closely with the key opinion leaders in the community as well. And through this process, we really are envisioning how we can generate the evidence also in this patient population. So with that, I'll pass on to Ala.
There we go. In the next couple of slides, we're going to look, is myotonia important to people with DM1? And it has been outlined quite nicely by Tanya about the complexity of the therapeutic approaches that it can take to address the needs and the pathophysiology of the DM as a disease. Right, I was trying to switch my slides. So as Tanya has outlined, the cascading symptoms that come with the condition as myotonic disorder or myotonic dystrophy type 1 and type 2 closely related to this spliceopathy that leads to all different manifestations that are presented here as the key features of the DM, and the major feature also is attributed to the skeletal muscle. So the next couple of slides, the data and the overview of the scientific literature that I'm going to share with you will encompass both type of the myotonic dystrophy, so the type 1 and type 2. And the symptom that we're going to focus today is indeed the myotonia, or inability to relax the muscles after the voluntary contraction. So on the right-hand side, if you look at the manifestation of the myotonia, which is only one symptom among all the different clinical forms of the DM1, you can see that it spans all ages of onset, and it's accompanied by the other symptoms very closely. So these are the data from the DM scope registry. That is the at the moment is mostly intensive registry of collecting data in France. And if we zoom in into this registry of the DM scope, what we can see is that the severity of the myotonia is the only symptom that is present in the different onset of the disease throughout the progression of the disease, unlike the other symptoms. So, for example, the frequency of the DM1 features from the top to the bottom would either decrease with different onset of the disease or increase while myotonia persists throughout different onsets of the condition. So what is that important to people with DM1? And we have several evidences that summarize this data in the literature. For example, the Christopher Project and PRISM that collected data based on the patient-reported outcomes in the form of the surveys. Altogether, this is more than thousands of people with DM responding these questions. And what we can say is that most commonly reported symptoms in overall myotonic dystrophy were the muscle weakness, fatigue, daytime sleepiness, and the cognitive issues. And these symptoms were also most impactful. Also, difficulty concentrating and learning difficulties were also important to patients with DM1 and DM2. If we zoom into the myotonia, what comes obvious is that there is quite often a misperception and the lack of awareness of impact and also of understanding of relevance addressing myotonia in people with myotonic dystrophy. So what you can see here is that in spite of a high prevalence of the DM high prevalence of myotonia independent of the onset of the disease, so in overall or looking into subtypes, so DM1 or DM2 or congenital onset of DM, the impact score is also somewhere moderate. But when we look into clinical data, besides the fact that the data are quite limited, we quite often have heard that there is a misperception of the presence of that myotonia and the way or the deepness to which the symptom contributes to the overall willingness to manage the symptom of myotonia in these patients. And so what we were interested in is to undergo some research with the help of the specifically developed survey or questionnaire to the patients, which we called ANSWER, because we really wanted to have an answer to this question. And it was around revealing the burden on daily activities in the patients due to myotonia. I will share with you some key findings from this survey. So it was over 386 adults living with DM, participating from over 23 countries globally. And the majority of these patients majority of these people were people who were suffering from the DM, and only 18% also there were carers who could also respond to the survey on behalf of the patients. As you can see, about 74% were with the diagnosis of the DM1 and 26% with the diagnosis of DM2, and a good distribution also between female and male. Some key findings. So here we were looking at the diagnostic delay in this patient. So asking to indicate the time of the age when the symptoms of the myotonia, not the myotonia, but the first symptoms have occurred to these patients, and the real diagnosis that they have received confirmed clinically. What comes out is that there is a significant delay in diagnosing these patients. For DM1 on the median period delay about 3 years, and for DM2 patients up to 12 years, which is really considerable. When we looked when these patients first seek clinical consultation, it turned out that for DM1 patients, it was the myotonia. So with 43% of respondents confirming that they were complaining about myotonia when they first saw a clinician for consulting. And for DM2 patients, it was the lower body muscle weakness. So over 75% of participants were experiencing myotonia at the time of the survey, with additional 18% reporting a previous history of myotonia. And this graph also shows the distribution of the participants who reported about having the myotonia separately for DM1 and DM2. And what is interesting is that when we looked at the stratification of these patients based on the age, so we looked in the younger patient group, less than 40 years, somewhere in between, and then older than 60 years, and then looked into individual symptoms of myotonia in different anatomical part of their bodies, it seems that independent of the age, the myotonia has persisted in terms of the frequency, but also without lower body muscle weakness reducing over the time or reducing over different age groups, suggesting that the myotonia is confirmative to the data that I've just shown to you from the DM scope, is a DM1 symptom that is not correlated with the disease progression. And you can find it throughout all different onset of the disease at different ages. And although there is a muscle weakness that is increasing with the time, there is also some natural history data that comes out that confirms that the myotonia and the strength are tightly connected. We don't understand the full picture yet, but it seems to be the case. And the ANSWER survey has also confirmed that. So if we look at the impact when we were asking the patients, next to the frequency of the symptoms, what were the most impactful symptoms that they were reporting. And for DM1, it was the hand and arm myotonia, upper body muscle weakness. and as well as the lower body muscle weakness. And for the DM2 patients, it was the lower body muscle weakness and also the leg myotonia. So both groups have also experienced muscle pain, daytime sleepiness, fatigue, and all these symptoms were seen at least weekly, as we remember also from the frequency. So when we asked these patients whether myotonia is important for the patients with DM1 and DM2, indeed, this symptom was coming out among the 5 key that we've outlined here. So for both patient populations, muscle weakness, fatigue, myotonia, balance problems, and/or falls, as well as the muscle pain, were among the symptoms that were most important for patients to improve. This one looks a little bit more complicated at the first glance, but here, when the patients were asked about the importance of undertake the daily activities as well as the decisions that they need to take in terms of their education and work, and also the adaptations that they need to put in place in their workspace, we've looked into the granular analysis of whether these changes were due to myotonia or not, or maybe even partially due to the myotonia. And we felt that there were some interesting findings as well. If you look separately for DM1 and DM2, mostly the limitations that were pointing out by both patient groups were due to the myotonia around the activities and around the active part, for example, the reduced ability to exercise certain job or continue in the training, or well, at least some of these were also partially to the myotonia, and it was to some degree also difficult to distinguish because of, again, the multisystemic manifestation of the disease. But we've limited this patient population for the analysis who have said before that they do have the myotonia. And then finally, in spite of myotonia being present in all the different subtypes of the DM patients, and as we have outlined before in the different manifestations as well, and being one of the symptoms that the patients wanted to improve, when it comes to the treatment experiences, majority of the patients, so up to 60%, said that they were dissatisfied or only moderately satisfied with how their DM was managed. Here, we can refer maybe to some of the cultural backgrounds. As you can see, there are different data that are coming out depending on the country that we would look at here, with maybe because of some clinical practice being established in some European countries versus US or maybe the rest of the world, or yeah, there is an example also of Canada, with Italy, Netherlands, and Canada maybe having a little bit higher rates for at least moderately towards being satisfied, or in particular also for France, a great portion of people being somewhat satisfied. But as a matter of fact, up to 60%, for 60%, it was not the case. So we are willing to take this work a step further, and from what I have been showing to you based on the Christopher project, where I've highlighted that quite often there is a misconception between the perception of the myotonia and the burden that it brings to the patient's daily activities and daily life reported by the patient, and how this is handled in the clinical practice. We have been looking into the opportunity to leverage the existing database that I have already mentioned, the DM scope in France, with French version of the ANSYS survey. So here, the DM scope data would allow us to look at the close correlation between the clinical myotonia assessment, the way how it's done in the clinical routine, because these are really real-world data, the disease severity, and also they use some functional tests as well as the educational level, with the way how the people responding to this questionnaire would provide their answers on the questions around their symptoms, around manifestation of the myotonia, and the burden that it takes on them. So we plan to enroll about 100 people to answer to this questionnaire, and we will be happy to share data on this study by the end of 2025. So to kind of wrap up on this part, looking into the different key disease areas and the treatment-related challenges that we anticipate in such a vulnerable population as myotonic dystrophy due to the myotonia. We've listed here a couple of aspects that are important to the patients that should have an impact on how we develop the management options, including for myotonia. And that has something to do with the disease on itself. So with 65% of patients with diagnosis of DM1 having experienced the dysphagia, so we need to take into account the difficulties to be able to swallow the treatments when it comes to the oral options, the cognitive impairment, that may affect the memory. So this is where we need to look at maybe reducing the frequency of treatment even during the day. So reducing that may also, as an outcome, reduce the adherence to the treatment for these people. Some level of the anosognosia that is prominent in most of patients with the diagnosis of the DM1 can also have an impact on motivation, having a motivation to adhere to the treatment. And of course, the existing pathology that is already established for some off-label standard of care solutions that is also reducing the clinical effect at therapeutic levels that would be mostly optimal for the people with DM1. And of course, the last but not least, the safety profile that is currently unknown for the off-label of existing anti-myotonic options for the people with DM1. Patients tend to discontinue due to tolerability issues. And we need to take into account all these options when we think about the development of the therapeutic candidates, but also when we plan and when we carefully design our clinical studies. So the study that I would like to present to you today is called HERCULES, and it is based on the clinical assessment for safety and efficacy of Mexiletine prolonged release formulation. The outline of the study is in a simplified way to just present it to you as a high level, but you can find more details also on the clinicaltrials.gov. The HERCULES study is a 6-month core study. It's a randomized clinical trial, which is also double-blind with patients being eligible from 16 years old with a confirmed genetical diagnosis of the DM1 or DM2. And what we look here is to include the patients who have the presence of myotonia that is important to them. So we will use a specific tool that has been developed for these patients to measure the myotonia presence, and we look for patients who have more than 3 seconds myotonia in hands. Of course, based on the profile of this medication, we will also look for candidates who have the normal cardiac examination. And the cardiac examination is a central core part of the whole study with a very detailed cardiac assessment at all study visits. So patients will be randomized 1-to-1 against placebo with Mexiletine PR, so Mexiletine prolonged release. And the drug will be up-titrated weekly to the therapeutic dose of 500 milligram. We have the readouts at weeks 14 and 26, when at the end of the study, everybody will be invited to roll over into 18-month extension study that would allow us to collect more data on the safety, but also long-term effectiveness of Mexiletine PR. On the right-hand side, there are also some key numbers on the patient numbers, and also that the study will only be going in Europe and UK for the moment. The primary objective, and I think I have said that already before, but I would like to reiterate, we're looking into the efficacy, but also safety of once daily Mexiletine PR treatment for the symptomatic management of myotonia in patients with DM1 and DM2. And we will be looking into, as an addition to the patient-reported outcomes, but also the efficacy on functional capacity, outcome measures, and general safety on the top of the cardiac safety of Mexiletine PR in this study. The PK, so the pharmacokinetics, will also allow us to make some informative decisions going forward for pediatric patient population and also for adapted pathology with Mexiletine PR in the future. So this is in a nutshell also for the patients, the information that you will find at the clinicaltrials.gov. Two codes also listed here for both studies, for HERCULES and ATLAS. You can check the website. It is not active yet, but it is becoming all the material and associated with the studies have also been already approved by the ethics committee. You can visit the study and see if you can qualify. And with this, I think we can go into the Q&A session. Thank you.
Thank you so much. That was great. We definitely have a few questions, and I think if folks would like to submit additional questions, just a reminder, you can do that in the Q&A button to open up the window and type in your questions. So look for the Q&A button. It's usually at the bottom of the screen, and you can type your questions in there. And you do have the option to send it anonymously, just as a reminder. Just make sure to check that box. Okay, we have one big question that came in directly to me, which is, if the study is in the currently in Europe, when or will it ever come to the US, and why isn't it here now?
So maybe I should take a stab at that first. I think that Ala already alluded to it, that what makes a medicine is the data that it comes along with. It's not necessarily the chemical structure of whatever it might be. It's that it has been proven to be safe for patients to take at a given dose, at a given frequency, and that it's beneficial for something that you need help with, basically, in your body. And our job is to do the trials and produce those data sets, and then it'll be up for regulators like FDA or EMEA or Health Canada to make a decision on whether they think this is sufficient to get it approved. And a lot of the times they agree, and sometimes they do not agree. And I think that what we see right now is that there is just not a complete agreement, and timing at least, that shows that a medicine is available or registered for an indication in Europe, but not in the US. It's not the only area at all, but that's just the lay of the land. So, to answer that question, it'll basically come down to what will FDA think about the data package, and will they approve it? That will decide that. Until that, even if the chemical structure or the generic molecule or product is available, it's important to remember that it's not indicated until it's indicated.
Okay, thank you. Appreciate that. Next question, we have a couple of them, is how is this Mexiletine different from the Mexiletine that is currently available and used primarily from what they can tell for heart indications?
To take a first stab is a little bit the same question, or the same answer I just gave. One is that what we are trying to do is to take an investigational medicine and test it in a way that could potentially give it an indication for use. And until it has achieved that indication, it is not available. So it's important to remember that the difference is not necessarily only in the chemical structure, if you think of it like that, but the difference is in the data package. So therefore, these will be two different medicines until something is approved for the M. It's also a different formulation. So as we are learning, we can learn on clinical outcomes, we can learn on safety profiles, we can learn on bioavailability, etc. So the learning has led to a formulation that we think could be more beneficial for patients.
Yes, there are some technical advantages also of the different formulation. The key features is once daily, if I can add, and also instead of the available form in a capsules, in a sachet that you can just have in a resuspended form and drink. So these are technically also different features of the formulation that is going into the clinical study.
Big question that we have received a few times during the registration process and now actually live. Folks are very excited to see a drug being tested for DM2, and they would be interested in knowing just a little bit more about, how or why do you think that this will work for DM2 versus DM1? What is the data behind that, or what do we know that would lead us to this point? There's a lot of data on DM1, but not as much on DM2. And so folks are wondering why and how, and what can you tell us?
Mm-hmm. Maybe I can even share one slide because we have seen this question before on the DM2 patients. So we are including patients with a diagnosis of the DM2 into our study, just based on a simple fact that although being a different disease, the overlapping symptom in this patient population is a myotonia. And although we do not know whether the drug is going to be effectively managing the symptom of myotonia, this needs to be tested in our trial. We have the patient population included into the study. So this will be based also on the data that we will obtain from the study.
Okay, thank you. That's very helpful. Regarding Mexiletine as a medication for heart, there are doctors who will not prescribe the existing Mexiletine, but if they have current cardiac issues, that is, specifically for myotonia, will this new version, this different formulation, be different in that respect, where you can or can't? Is it going to impact cardiac in the same way? What do we know, or how will we be able to think about this differently?
Yes. So Mexiletine, although being actually a previously approved anti-arrhythmic drug, can also paradoxically induce the proarrhythmia. And that's why close monitoring of the use of Mexiletine in particular patient populations such as DM is very important. So the study design of HERCULES is as such that we very closely monitor the patients at each dose escalation as well as at each study visit. We have detailed ECG and also 24 hours Holter. And as of today, there is a accumulating body of evidence looking at, including some trials that have been done previously in US that have looked at the cardiac safety in respect of long-term use of Mexiletine in this rare disease population, yet a full benefit risk profile has not been fully established, and our HERCULES study will also look into that.
Okay.
So I think that, as disappointing the answer might be, is that we don't know until we know. But I think the encouraging thing about what Ella just talked through is that there is progress on collecting those data, and therefore there hopefully will come a time where we get clarity on it. But what the outcome will be completely driven by the data.
And could you remind folks how long do you anticipate this study going, and at what point, if it goes well, do you think that it will be available and on the market?
I think it's $1 million question, and I will restrain from answering it, because if I was that good, I would be a betting man with great success. Drug development and doing clinical trial is a challenge, and we obviously work with planned timelines, but these are internal objectives that we work towards. And I think that it will be wrong of us to give anyone a false hope, or a hope that is not being justified. So I'd rather say that it will take the time that it will take. But what it is worth, I think you can trust that we want to make it happen as quickly as we can without compromising quality.
Okay, let's hope that that moves smoothly, the whole process. We have 2 questions about slides, if it's okay to sort of go back to those. One, slide 18, there's a question for clarification on whether that was people who are not satisfied with their DM treatment that you're speaking about, or people who are not satisfied specifically with their myotonia treatment.
It was specifically to myotonia. We did not formulate the question specifically or explicitly putting out myotonia, but patients were asked before about having the myotonia, and only those who have said yes at the moment, currently, because we've analyzed the data separately of having had myotonia before or experience it currently. We've only then asked those patients who said yes, how satisfied, if you have received any treatment or your management, how satisfied you were. So it does relate more to myotonia specifically.
Great. Okay, that is helpful. Thank you. And then another question that came in from slide 20, there is the use of dysphagia, the word with an S, which is typically language difficulty, versus dysphagia with a G, which is difficulty swallowing. And so asking for clarification of which one you were referring to.
Swallowing, apologies. Swallowing, yes.
Got it. Okay, thank you. There is another question. Curious if you could maybe provide some insight. This is a big question and a little complicated perhaps, but why wouldn't the FDA or another drug approval agency allow clinical trial to move forward or certain research to be done? This is much bigger than just for lupin, but if folks are wondering sort of why isn't this coming to the US, or what's happening and why you choose to go to Europe or anywhere else first, why is it that you would start there versus somewhere else, or what would be preventing the FDA from moving a trial forward?
That is a big question. I think that drug development is highly regulated in most places, certainly by EMEA for Europe and FDA for the US. And they have criteria for what they would like to see. what they would want to see from a safety perspective before they allow it to progress. There's different stages. Some of you might be well aware of, animal studies coming first, toxicology studies, eliminating any significant risks. Every drug would go into healthy volunteers first to see the reaction before you will take it into patients with a diagnosis that needs to be treated. So I don't think that we can answer the question now. We can just say that it's a very regulated process, and rule number one is to protect patients, safety first. And then subsequently, if there are signals that this might be able to help patients with a certain condition, then you test that progressively without putting any human being at risk that really can be avoided. The burden of evidence is described by each of the agencies, but it's always a judgment call, and judgment calls falls out differently from time to time.
Okay. I think we may have answered all of the questions that have come in during the presentation. We'll give folks just another minute. If you have any other questions, to please go ahead and submit them in the Q&A through the Q&A button. Just give folks just another minute.
Maybe I can make a comment, if okay, while they are thinking about the questions. And basically, thank you for the work that you do. And it's not just a superficial thank you. You probably noticed that we spent quite a bit of time in the presentation and the slides about understanding the burden on patients. You can do that at an individual level. That's really important. But also doing it as a disease level where the frequency and the severity of patients in general is important. And we do that because it's very easy to be derailed and pursue things that are possible, but if they're not helpful or if they're not addressing the real problem, afterwards it's only going to be half the help that it could have been. And therefore, the in-depth understanding of what problem is it really that we're trying to solve is very, very important. And the work that you do is super helpful in progressing that agenda. You're a great dialogue partner, getting into contact with the right people, but also the passion and the energy that goes into defining that is actually leading to better medicine development in our view.
Thank you. Appreciate that. So we do have, it looks like, a couple of other questions. If you had a crystal ball and this drug gets approved, will it be available for children, or will there have to be another study?
So if I would have to answer that question, I probably would get in trouble, and I will take an answer this way so that my colleagues don't get in trouble either. We obviously would like to get as many patients helped as possible. And especially when you work in diseases that are not that common, in rare diseases or ultra-rare diseases, the judgment call by an agency like FDA is really important. I think that they would be willing to extrapolate data that you have, or they're willing to work on small data sets to the degree that it is meaningful, then it is safe for them to do. And therefore, in a situation like this one, you need an even bigger crystal ball to get close to it. So it's important for us to stick to what we know and what we can prove. I worked with a colleague in IND who I will quote for saying once, in God we trust, everyone else has to bring data. And, our job is to bring the data, and we will do our utmost to do that, and without any delays that can be avoided. But it has to be a data-driven decision, and therefore it will have to be answered later.
Okay, thank you. Question about potential side effects, if there are any known or expected, including anything for the GI system. So are there any upset stomachs with this form or the current form of Mexiletine?
So the new formulation that goes into clinical study is designed in a way to reduce the tolerability issues, but of course this needs to be tested in our clinical program, including the GI irritation and the reflux that is commonly known for Mexiletine, but also the improved distribution and also the PK profile that covers basically the daily need for myotonia control should also bring product profile or the treatment profile that should be at least comparable to the previous version of the Mexiletine, and of course the safety next to the cardiac safety are the key points in the current study and in the current design.
Thank you. Let's see. We have another question. Just sort of as the regulatory process works, if you would be kind enough to just share a little bit about why healthy populations are studied before people living with the disease. If people living with the disease are willing to take the risks to see a treatment come to market, why are we studying healthy folks first?
I think it's a big question. Probably FDA is the one to answer it. But if I can give you a little bit of a layman version on it, I think that if you give a potential medicine to a few individuals who seems to be healthy as far as you can tell on all parameters, then you will have a better chance of see signals of potential side effects or effects that you want to make sure need to be aware of. Remember, we obviously develop medicines to help with a condition, but rule number one is safety first. So therefore, we need to rule out any unsafe elements before we pursue that. I think that's kind of in short the golden rule.
Thank you. Are you aware of any research about crossover in research potentially involving dystonias and myotonia?
This is really an interesting question, and maybe I just read this in the chat. I would like to invite you to a Dystonia Europe webpage. We have worked with this patient advocacy group and left comment, not a comment, how do you say that? That was actually developed in collaboration with one of the professors of and chair of neurology from Milano, from Italy, to dissect a little bit the difference between dystonia and myotonia. So it's called, the rubric is called not dystonia because sometimes it can be mistaken for myotonia and the other way around. But I think it's quite clearly laid out on that webpage of Dystonia Europe. Please go and, there's a lot of information, including from this key opinion leader of neurology that is left there to explain.
Thank you. I think we really only have one question left, and that is, it's a perfect question to sort of wrap us up, is if this is all going well, when might you have some data to provide an update in the future?
Oh, that's a million-dollar question. I don't have the timeline in front of me right here, but I think you will have to count it in years, unfortunately. So again, we will push this as fast as we can without compromising quality in any way or form. But it'll probably be a couple of years out.
Okay. Okay. I think we have answered the essential questions. Thank you all so much for being here and for the thorough presentation about Mexiletine. We're very hopeful. We're looking forward to seeing progress, and hopefully you will be back in the near future with some updates. Um, if you, for those who are still with us this afternoon, if you like today's webinar, please consider donating and helping us to further the support for MDF and our mission. Please also consider joining our registry. They're extremely important to support our clinical trial and study activities. Without those, we cannot connect you with trials and studies. So please register. And thank you again to the Lupin Neurosciences team, to everyone else who has joined us today. We wish you all a very happy holiday season. Thank you.